Is it working?
Yep, gotcha.
So I would argue from the Genesis starting point, okay, we can make predictions, we can make retrodictions. So the Bible claims that we are created in God's image. Okay, so there must be something about us that reflects the divine. And so the prediction would be, okay, maybe we can get a sense for how God designed the biological world based on the way we design things. And it just so turns out that we design in, again, homologous patterns, nested hierarchical patterns. We design interesting mosaics, transitional like vehicles. We have this interesting field, biomimetics. Okay. And so all of this is not by design in the sense that human engineers are designing modes of transportation in a way that supports the creation model. No, they're designing that way, and it just so happens to turn out to match the biological world. And so that is confirmed expectations from the biblical creation model. And I want to relate this to ERVs, and then Mark or Grayson, take as much time as you want to respond. This all comes down to function. And so ERVs were brought up earlier from Mark, if I'm not mistaken, with Kent. Okay, why do we share endogenous retroviruses and ERV-like sequences with primates, with chimpanzees, basically the biological world in general? Well, we have paper after paper. This is from 2021. They admit long disregarded as junk DNA. And so they're admitting that evolutionary scientists used to look to endogenous retroviruses as being junk, genomic dark matter, non-functional evolutionary leftovers. But now what they're finding more and more, again, this is 2021, they have turned out to represent important components of the antiviral immune response. They not only regulate cellular immune activation, but may even directly target invading viral pathogens. And last thing I'll say, what's interesting is the specific makeup of the ERV, why they share sequence similarity to exogenous retroviruses like HIV, right? You got the LTRs, long terminal repeats, GAG, POL, and ENV genes, or components of the ERV. Those components, the structure and build of the ERV is actually necessary for them to carry out many of their roles, two of them being this, fighting off exogenous retroviral infections, and tumor suppression. They do that through viral mimicry, which means they have to look like the viruses in order to mimic them and therefore fight off tumors. And so long story short, the reason why we share endogenous retroviruses with chimpanzees and other creatures in the biological world is for common design purposes. They serve a function. They serve a benefit. And they have to look the way they do to serve that operational role. Go ahead.
Okay, so you went through a lot there, actually. So this whole idea of religious belief kind of thing, you basically start with a religious belief.
Yeah, I was going to ask if you could zoom in on it. There you go.
Yeah, you basically start with a religious belief and then try to fit all of the evidence to it. It's not, sort of, misunderstood or anything. That's what you do. You're basically cherry picking. This is your paper that you brought up. You had a reference for it down the bottom. I'm so glad you brought it up, Donny. So when you say they're necessary for the biological function of that animal now. It has become necessary. Like when you say they're necessary for life, they're not necessary for all life because there are other life forms, other species that do not have these viruses. But what's interesting is these viruses match perfectly to the hierarchical tree that we have of evolution. And now this is your paper. This is the one you cited. It's basically telling how the genus that are indicative of a long-lasting coevolution with endogenous viral pathogens and the simple yet effective antiviral mechanism that you're talking about that has evolved several times independently during mammalian evolution. All of this is in the paper that you are quoting, how it goes into evolution. It quotes how we know that fusogenic human endogenous retrovirus envelopes identifies syncytin-2 as a gene conserved on primate evolution. So all of this is due to evolution. That is well spelled out in that paper. It's just you choose to ignore that which you do not want to fit with your religion. It does not support independent creation at all. It supports evolution. This is what the paper says. So while you can cherry pick out of papers all you want, you are misrepresenting the paper, because they quite clearly tell you that evolution is responsible for these antiviral productions. So, you know, I don't know why you're using this paper that 100% disagrees with you, but, you know, it's absolutely shocking that you will sort of use this paper at all.
Okay, let me respond. Leave the paper up so we can examine it. Okay, so high on rhetoric, little on substance, which is typical with Mark, my boy. And so I do love you for that, though. You said you're shocked. I'm flabbergasted that you're repeating the same points that I've refuted over and over again. Okay, so Mark, unfortunately, doesn't know how to examine these papers in a way that recognizes unvalidated assumptions and guesswork and the actual empirical data. Okay, simply asserting these functions were co-opted does not demonstrate true co-option. Yeah, you'll see in these papers that I frequently cite that these are motors of evolution and they were co-opted without actually demonstrating true co-option, the true acquisition or evolution of these functions. Okay, acting as antiviral protectors, acting in cell stress responses, functioning in embryological development, where literally if we did not have these retroviral-like sequences, we wouldn't exist. Mammals wouldn't exist today. And these are just, I'm almost done, Grayson. These are just a few of the many functional roles associated with ERVs. And again, the major question from more informed evolutionists is, why do endogenous retroviruses share sequence similarities to exogenous retroviruses? Mark dodged, duck, dip, dive, and dodge. He did not respond to the point that these ERVs require their very specific structure, their makeup, LTR, GAG, POL, ENV components, to carry out at least two of their functional roles. And so they have to look like the viruses, the exogenous retroviruses, to carry out at least two of their roles. And so I'll yield there. Go ahead.
Okay, I just want to make...
Well, hold on, Mark, because Grayson has had his hand up and he's been trying for a long time. Grayson, go ahead. And then, Mark, I promise you can answer.
Okay, mine is just very quick. It's just an actual understanding of fractions is enough to debunk all of this. If you just understand how fractions work, then you understand the flaws in all this reasoning. Donny is bringing up known examples of ERVs that have some functional component. But all you have to do is ask, what percentage is that out of the total number of ERVs? It's the same for all of these various forms of non-coding DNA, like long non-coding sequences, like retroviruses, transposons, like pseudogenes. The percentage, like you can find functional examples of each of these. But the question is, what fraction does that represent of the total? And it's a very, very small fraction. And if you understand that, you understand why the argument from Donny's side is not valid. Mark?
Okay. No, I'll respond. And we're going to go one point at a time. Mark can respond to my points, as I know Mark does, because he's an excellent debater. He's got all my points written down, and he's ready to demolish them.
Also,
So
Donny, try to get praise in with you too. You guys can take them on. It's not fair that you have to take them on single-handedly.
No worries. Maybe what we can do is I'll respond here, then we'll let Mark, who I always enjoy engaging with, we'll let him respond, and then we'll let praise.
Sounds good to me.
So if you could share my screen real quick, please, and then cue me when the screen is shared.
We've got it. Increasing knowledge.
Battle boy. And so this whole argument from fractions is very, it's premature. It's old news. Okay, we understand that at least 80% of the genome is active in terms of biochemical activity. Grayson would argue that this is mostly just noise, but this argument's easily refuted, and we can engage that if we have to. We've already engaged that for hours and hours. And so within that 80% activity, genomic activity, guess what we find? Pseudogenes, ALU sequences, endogenous retroviruses. And so that tells us that these are active. But again, we are in the beginning stages of understanding genomic functionality. Okay, we are just starting to recognize good design. We're recognizing these amazing functions in these DNA elements and units that the evolutionary community has acknowledged. They used to believe were junk. And so here you'll notice from 2021, same paper, our knowledge about endogenous retroviral elements involved in cell immune responses is what? Constantly increasing. And so when you actually dig deep into the technical literature, they are admitting that over time, our knowledge in terms of ERV function is increasing more and more and more. They've even now discovered that these transcriptionally silent ERVs have an advantage to the host and are functional. Many evolutionists have argued, well, these transcriptionally silent ERVs, you know, they must truly be junk. They must truly be the ancient leftover remnants of past viral infections. When in fact, no. Again, same paper, 2021, they talk about how the presence of these very specific ERV elements may promote recombination processes that can help with genetic diversity, help with adaptation. And so transcriptionally silent and also ERVs that are transcribed, they have a functional role, and they admit that the knowledge in terms of these functions is increasing more and more and more. And so the last thing I'll say, science is about making testable and falsifiable predictions. And so me as a biblical creationist, I can look at this data, this accumulating data and knowledge, and I can make very specific predictions. And those predictions involved DNA functionality. I predict in the next 10 years, we're going to find even more functional roles and abilities for ERVs, ALU sequences, pseudogenes, and just the non-coding regions of our genome in general. And so I'll wrap it up there.
Okay, so this is really a disingenuous argument by Donny, and it has absolutely no credibility behind it whatsoever. He's basically just saying, hey, because they have functionality, it must be something created, and that's just a facile argument. When he says in fact, it's not facts. That's not what anybody actually says is a fact. The thing is that basically what he's basically doing is saying, hey, if they have functionality, they must be designed. And that's a poor argument. We understand how they came. This whole idea that, oh, we get more information in science, so we understand more about these things that we thought were junk. It's like saying, hey, we understand more about cancer, so all cancer research is bunk. It must have been designed. It's a terrible argument. All levels of science, we're getting more and more information and updating our information. That's the difference between science and what Donny is using, which is faith. He basically has a belief in the Bible first, then he tries to fit everything with the Bible. And as Grayson pointed out, all you have to do is look at statistically how these have mutated. And we have a lot of information about them mutating. The paper that Donny keeps bringing up has references to show how these mutations can cause benefits in the cells, and we've demonstrated that through experimentation. This isn't not understood. And Donny has nothing to back up his claims that this was a created design. Nothing at all besides his own rhetoric. All he does is just repeat it. Oh, it has purpose, therefore designed, or it has function, therefore designed. And that is a non sequitur. Just because it has function doesn't mean it couldn't have evolved that function. And it is much more likely, if you listen to Grayson and the papers that he's using to show that the mutations in those ERVs produce benefits, just like any other cell in the body, it's much more likely to be evolution than some kind of created design. Which, and I'll just reiterate, the entire evidence Donny has for this is his religious belief. He's just asserting it. It's not fact. And there's no papers on it. There's no demonstration of it. He is just asserting that a creator designed these things to look like viruses, to have all the structure of viruses, but in fact, they're designed by something. If you can show that, then do so, Donny. Don't just assert it.
So let me respond. I did see Praise Unmute. We're going to get you in on this soon. Mark said he wants to reiterate, and I appreciate that, because his entire rebuttal there was one massive reiteration of talking points that I've already demolished over and over again. And this is why I love Mark. I do appreciate him. And so he dodged my predictions, dodged my responses, completely dodged my explanation for why endogenous retroviruses share sequence similarity to exogenous retroviruses. This is necessary for them to carry out their functional roles in fighting off viruses and also in tumor suppression. He's also failed yet again to provide evidence for the co-option that he continues to assert. All we find here from Mark are claims and assertions, and Mark's responses here are a gift. They really are a gift to creation science, because they demonstrate how powerful the evidence is for the common design model, and just how lacking the evolutionary community is in terms of providing explanations, empirical explanations for these amazing functional roles. Just asserting co-option, claiming co-option, as we see in these papers, it's really not going to cut it. I want to see testable and falsifiable predictions being made by Mark. If you could share my screen real quick, because that's the gold standard of science. And that is what I have done in this back and forth, is I have looked to the data in these technical papers. This is what Mark doesn't understand, is we have the data presented in these technical papers, okay? So we find that these ERVs are incredibly functional in determining cell types, regulating genes. Basically, without ERVs, and this is where I want to find a very specific paper. I've got an entire presentation here at 200 slides. They admit in the technical literature that without ERVs, we would not exist. Mammals would not exist. Notice right here. Okay. Without these very specific HERVs, human endogenous retroviruses, humans and mammals would have never existed, because ERVs are necessary for the placenta and also for embryological development. In terms of the tumor suppression, it's very interesting, because transcribed ERVs can actually mimic an exogenous retrovirus, a harmful retrovirus. And as a result, the immune system, which is joined by the P53 guardian of the genome, this incredibly complex protein, P53 protein, and they target that very specific cell with destruction, because it looks like it's being invaded by a retrovirus. And this helps to fight against tumors. But again, the endogenous retrovirus requires those similarities to exogenous retroviruses to carry out that very important role. And so I like to exist. Mark likes to exist. Why do we share endogenous retroviruses? It's because we require them to live. It's the same reason why all types of modes of transportation have an engine in common. Okay, because without the engine, those vehicles are not going to get very far. And so I'll yield there.
If I can just really quick point one thing out, and then Mark, I'd like for you to get a better response.
Guys, we also, well, Based, you can go, and then we'll let Praise get in, because he unmuted forever ago, and then Mark, you can get back in. So
I just have something really quick to point out. Donny is pointing to like 1 or 2 specific examples of ERVs with known function, and he's trying to extrapolate that to say, see, from these small number of examples, we can know that all these millions of ERVs have function. That is logically fallacious. You cannot make that extrapolation error like Donny is trying to do. And he's trying to like smuggle that in with all this rhetoric. Like, see, look, we need this one ERV. Therefore, all ERVs are necessary. That makes no sense. Praise, you can go.
And I'll respond to Grayson after
Donny,
Praise.
it's Praise's turn, and then Mark.
Well, that's what I said. I'll respond to Grayson after Praise, but I do got those points noted for me to
It's
respond.
Mark after Praise, and then you can go.
Right.
So I just want to bring up with junk DNA is the C-value paradox, if you guys are familiar with that. So for a long time, evolutionists have thought, well, there's genomes that have, extra size or large genomes out there. And that would account for the junk DNA. But however, they found that it really shows that genomes are more than just coding DNA, and this non-protein coding DNA is their reason. So it's just begging the question. So I'd like to see how Mark or Grayson would address the C-value paradox. I'll end it there.
What
You're
question
muted,
we're begging? I don't know what you're talking about with C-value, and I don't know what we're begging the question of. That makes no sense to me. But I don't need to. I just address Donny because I don't need to point out why endogenous retroviruses have such a similar structure to exogenous viral pathways because his paper does it itself. The paper that Donny keeps referencing does that. It explains the importance of FV1 in antiviral immune responses is further highlighted by signatures of positive selection. This is natural selection in the FV1 gene in the genus mus. That's the genus of the retrovirus that are indicative of a long-lasting co-evolution with exogenous viral pathways. So I don't have to point out why he's absolutely wrong about that. His own paper points out why he's exactly wrong about that, which is, I'm sorry, Donny, that's hilarious that your own paper tells you you're wrong and you keep referencing it saying, why is it so similar? And your own paper tells you if you read it all the way through. This whole idea that because it has a benefit, then humans wouldn't exist without this retrovirus. Yeah, we wouldn't exist without a lot of things. We wouldn't exist without the ability to breathe. We wouldn't exist without this. We wouldn't exist. Like the whole idea that, it wouldn't be humans wouldn't exist without this thing. Yes, that's correct. That's how we evolved. That's not anything in the favor of individual creation. All it's saying is, hey, the way that we evolved, we had these retroviruses that helped us. So we co-opted them, just as his paper says. So there's no argument here. All of this is rhetoric and sleight of hand to say, hey, and the whole idea that there wouldn't be organisms if there was no, say, for instance, the placental retrovirus, which I think is the SIRH family of retroviruses, that isn't true either, because there are animals without that family of retrovirus. There's monotremes that still lay eggs. They don't use placentas. There's marsupials, which don't use placentas either. They use a Joey system. So this assertion that humans wouldn't exist, yeah, it wouldn't be humans, but it'd be something else. Because if the retrovirus didn't exist for, say, the SIRH, we wouldn't have placentas, but there would still be something. It's just saying, well, if we couldn't stand on two legs, then we wouldn't be humans. Yeah, no joke. That's obvious. So I don't know what Donny's point is here, but it's absolutely failed.
Okay, if I could screen share real quick, Stephen, my brother.
You're up.
Okay, so firstly with Grayson, my second, maybe third last response was directly to the argument he just repeated. And I've shown directly in the conventional literature that both transcriptionally silent and transcribed ERVs are functional. I've pointed out how the knowledge in terms of ERV functions is constantly increasing, not decreasing, but increasing. And that's why we as biblical creationists, we can actually honor the gold standard of science by making testable and falsifiable predictions, in this case, specifically on DNA function, as we would predict, since the trajectory of discovery really does favor genome-wide functionality, is more and more functional roles for these ERVs. But I pointed out earlier that they've discovered over 80% of the genome is active to some extent. Okay, transcription requires ATP. It's an energy intensive and resource intensive process. Every time you add a single nucleotide to a growing RNA chain, you're consuming energy. You're consuming ATP. And so this tells us that that activity is not spurious. It's not just noise, but it's real beneficial activity. Transcription-wise, at least 60% of the human genome is transcribed. But within that 80% activity, we find all of these ERVs. And so that tells us that the majority of ERVs are active and therefore functional. Mark, still a lot of dodging, a lot of assertion making, a lot of claims, not really addressing anything. And I've got an entire presentation here based on that paper. So yes, I've read it, I've examined it, and I've presented on it. And so they go through the various components of an ERV, like the LTRs, which stand for long terminal repeats, the ENV, the gag, the pull. And they talk about how each component acts in this functional ability for the ERV to fight off exogenous retroviruses. Okay, so right here you can see this excerpt on the LTRs. Over here you can read about the ENV proteins and how they directly inhibit viral infection through a process called receptor interference. And so you can go through this entire paper for yourself, and it provides a very elegant answer to why ERVs resemble or share sequence similarity to exogenous retroviruses. And it's because without these similarities, they would not be able to carry out their functional abilities. But the fact that they are functional points us to common design rather than common descent, especially for the fact that we have yet to see any evidence for co-option. How did these ERVs evolve the ability to act in embryological development to the point where if we did not have them, we literally would not exist? In the same way that if you took your engine out of your vehicle, you're not going to get very far in that vehicle, because that vehicle requires an engine in the same way that we require these ERVs. And I'll wrap it up there.
Mark, where did you go?
Did you want to go, Grayson? I was muted for some reason. I'm not
Okay,
sure
I was
why.
going to ask if you were muted, but
I'm muting everybody when they're not talking.
That's okay. That's all right. I just wanted to ask Grayson if he wanted to go, or whether you want
Also,
me to
Mark,
step in.
you had two screens shared for a moment. Did you remove
Yeah.
the one you don't want shared? So the one remaining you
yeah.
want
Sorry,
shared?
my bad. My bad. Yeah,
I'll
sorry.
just real quick point out that in that quote that Donnie was pulling from that paper, it said the LTRs mentioned above. It's not referring to all LTRs. It's mentioning the one or two that were brought up in the paragraphs above where he mined that quote from. So again, nothing he showed is showing that all of these ERVs are functional or even implying it. None of these authors are trying to imply what Donnie is trying to imply from their quotes. Mark?
Yeah, so this is the thing, and I didn't dodge. I pointed out in his own paper that sort of, there's nothing that I have dodged at all. This is a paper for rapid evolution in F1 in animals. If you could share my screen now.
I don't see anything shared, Mark. Oh, I'm sorry. No, actually, no. You have nothing shared.
I have nothing shared. Okay, well that's my fault then. Hold on 1 sec. I'll try it again. You're right. You're absolutely right. OBS was a nightmare. I'm just going to say that right now.
Dude, I'm telling you, I worked in tech support when I tried it. It sucks. Oops. I mean, it's great, but it's complicated.
Yeah, so this is the reference they give for how they know that the mus group of retroviruses is actually, like, they have traced the phylogeny of these viruses themselves, because as a series of viruses, we know they evolve as well. And this is the sort of arms race that is happening between endogenous retroviruses and exogenous retroviruses, ones outside the body. This paper, which is referenced in the paper for switching sides that Donnie keeps referencing, is Ancient Evolutionary Origin and Positive Selection in Retroviral Restriction Factor for FV1 in muroid rodents. So that's the one that we're talking about in switching sides. Now, it makes clear that this retrovirus has a long history before it even became the mus series of retroviruses. So here it's talking about the rapid evolution of FV1 in African murids. Basically, it says that shows signatures of positive selections in genus mus. Our present analysis has shown that FV1 has apparently retained full coding potential for millions of years before the appearance of the genus mus. We extend this positive selection analysis of FV1 to include other clades in the family Muridae, and you see the family Muridae of retroviruses here. So it basically, they have linked this genetically to all kinds of other retroviruses as they become exogenous outside the body and get passed horizontally, and as they get endogenized and are passed vertically through the generations. So this paper, which if you follow the citations, it actually shows them mapping the genetics of these retroviruses into all of these species. And they're mapping the genetics of these retroviruses between species of viruses, which is fascinating. And genetically, it shows why they're so similar to exogenous retroviruses. It's because they're related to exogenous retroviruses. There is nothing besides Donny's assertion that this was designed and put in. And as Grayson said, like apparently they were designed and only a tiny proportion actually had anything, any functionality to them for some reason that Donny won't identify. I'm dodging nothing. I'm showing the actual papers, not just the original paper that Donny showed, but also the papers that they reference to show how we know these are not created instructions, but instead they are evolving viruses, organisms themselves. They are evolving, and we can track that through the genetic structure. Genetics is the nail in the coffin for creationism. And all Donny is doing is just saying, hey, if they have a function, then they must be created. That is not true. That's a non sequitur. There is nothing pointing to that. He won't show any evidence of that. He just keeps claiming it.
Before I respond in great detail, can you put that paper back up, Mark, real quick?
It's up, Mark.
Yeah, I guess so.
Can you show me in that paper where they've demonstrated an exogenous retrovirus infecting or infiltrating a host genome in the right way where they're passed on vertically? And from there, mutations wipe away any harmful effects or aspects of that retrovirus, and then it evolves the ability to function in either embryological development or in fighting off exogenous retroviruses or even in tumor suppression. Where have they observed that? Does this paper provide that data?
You're asking for something that happened millions of years ago.
Exactly. It doesn't.
The amount that this is, excuse me, the amount that this, excuse me, the amount that this is spread out. This is the thing. But we've seen that happen with other retroviruses. This is the thing that you don't want to talk about. We've seen that happen with the AIDS virus. We've seen that happen with other viruses.
Is AIDS an exogenous?
You want to just say, hey, all of that's coincidence.
You don't like to be cross-examined.
Excuse me, can I talk, or is this just talking?
Well, are we just going to have uninterrupted responses for 4 hours? I'm trying to ask you basic questions about the paper that you provided. Is HIV an exogenous or endogenous retrovirus?
Exogenous.
Exactly. So where is the evidence of an endogenous retrovirus evolving the ability? Show me in this paper, but you just admitted, well, it happened millions of years ago, so we can't. Exactly my point. You are asserting co-option. You are asserting and claiming that these endogenous retroviruses evolved these abilities without actually showing us empirically how this happened. And so again, I'll ask you, show us an endogenous retrovirus gaining the ability to perform in the embryo.
Johnny, can you show me Pluto going around the sun?
I can do that. I shared my screen of a paper.
No, I want Donny to show me Pluto going around the sun. I want you to show me that.
We can show you observations simply through telescopes and measurements.
Correct.
And all types
yes,
of ways.
We can show you observations. Thank you. Thank you.
So I rest my case there. I trust the audience.
You have no case.
Your lack of.
There you go, Donny.
Answer.
There you go. There's your answer. You happy?
No, okay, you could send me that paper, and I will bet you 1 million IOUs that this has not showed us in the lab an endogenous retrovirus going from non-functional to something functional in the embryo, to where if we did not have it, we literally couldn't survive. Are you
all right? But I'm going to feel really bad bankrupting you once you have to pay up on that bet.
So, co-opted ERV. Zoom into that.
Zoom in. On what?
To the highlighted part.
Yeah, like, can you make your screen bigger for us? There you go.
In some co-opted, ERVs, so right off the bat, they're assuming co-option, stem
Nope,
from...
they're showing it. They're not assuming it. They're showing it in their empirical measurement design experiment here.
Stem from genetic escapees that have lost vital sequences required for both TRIM28 restriction and
autonomous retrotransposition.
Okay, so they're observing this retrotransposition, autonomous retrotransposition in real time, and they are gaining the ability to what? Which function?
They are having an enhancer function of a 47 base pair sequence within the U3 gene. It's right there, that LTR. They're showing it the enhanced ability there when they've lost specifically their viral pathology by incorporating into the host genome.
So in this
there
paper,
you go. Pay up $1 million
IOUs. No, because still there's a lot of claims here in this paper. So they've observed in a lab the loss of these viral components in the LTR, where the LTR has now gained this enhancer ability, or the LTR already has the enhancer ability, and now they're just theorizing how this could have happened by looking to other LTRs and...
No, look, it says right there that the escapee, as in like the viral genome that diverged from the norm and incorporated into the host genome, that escapee conveys an activating effect on nearby neural genes. So it is showing a co-option of an ERV that passes on a functional benefit. Exactly what you said couldn't be shown, and you bet $1 million IOUs.
Not in real time. It's not observing this in real time.
It is.
And this is just an LTR. Experiment. This is just an LTR.
No,
we're talking about...
like, you have not read this experiment, Donnie, because you wouldn't be making this argument if you had. They are literally showing it in real time in their experiment with like the ERVs and an embryonic stem cell for a neuroprogenitor cell. They are doing this in the lab and showing it in real time. Pay up.
Okay, I will predict that's not the case, but if you could share my screen and I'll go over the LTR data and why this would be a flop regardless, because Grayson earlier said that, well, in this paper, it's just talking about those specific LTRs, long terminal repeats of the endogenous retrovirus. Well, here's the thing a lot of people forget or just don't understand, is that almost all of the so-called endogenous retroviral sequences in the genome actually represent just solo LTRs. And they've discovered, there's many papers describing these functions. Here's just two. They've discovered that they act in gene regulation. They can act as promoters and even serve as transcription factor binding sites. Okay, and there is several papers that talk about the functional nature of these LTRs. And so why are we to believe that these solo LTRs that have functional roles in the genomes of living organisms, they lack the GAG, the POL, the ENV components of an ERV? That paper you showed, that's what it looked like. They were just looking at solo LTRs, but I'll have to read the paper. I could be wrong on that. Okay, so why are we to believe that solo LTRs are necessarily the remnants of ERVs, where basically at one point they were full ERVs, right? LTRs, GAG, POL, ENV. But over time, through mutations, they've degraded to the point where now they're just a solo LTR. Well, no, again, we know that they function as promoters and serve as transcription factor binding sites. And so, yeah, we share just thousands and thousands of these solo LTRs with the biological world, specifically primates and the great apes. Okay. But for common design purposes, because of their functional ability and important functional abilities. And so it is the evolutionist that needs to demonstrate that these solo LTRs really were once just one component of a greater ERV unit. Okay. When it actually comes down to full ERVs, where it's not just the LTRs, but it is the ENV, the GAG, and the POL, this is where we find functions in embryological development. And that's my question. I want to see a non-functional endogenous retrovirus going from non-functional to something as incredibly functional as embryological development, where literally if we didn't have these ERVs, we wouldn't survive. Mammals wouldn't exist. Okay, so Mark's basic question is, why do all modes of transportation share engines in common? Well, it's good design. Engines are necessary, hearts are necessary, and endogenous retroviruses are necessary to sustain healthy life processes. And so, of course, I would expect all of the biological world to share ERVs in common. It is within these papers that we find their functional roles in acting in tumor suppression and fighting off exogenous retroviruses. They do this because they do share sequence similarity with exogenous retroviruses. That's the data. What's the interpretation? Well, the interpretation is they have those sequence similarities in order to perform their functional roles. They have to look like that which they are fighting against. That's basic viral mimicry. That's how that works. And so I'll wrap it up there.
All right. Well, now that we've got that gish gallop to try to avoid what we were just talking about and moving the goalposts, I would just like to tell Donny that I will accept checks, money orders, or Venmo or PayPal. Thank you.
And I would just like
So
to
you are
add
saying it's
something.
more than just an LTR that we're looking at in that
Yes,
paper?
it is not just an LTR. It's a total ERV genome. Yes.
Okay. Send it to me.
And
I'll
I
look
would
it
just like to add something that's sort of important for me. That sort of this idea that you know, you're sort of saying, hey, show me the ERV being actually fixed. And Grayson showed, you know, that paper. You've got to send that to me, Grayson. I love you. That's amazing. And like, but the whole idea is you're claiming that they were created by some sort of God deity thing. Can you show us God creating this thing and inserting it into the genome, please? Why don't you live up to your own standards?
God creating gives us observations and implications to where we can make testable and falsifiable predictions. That's the gold standard of science. And that's what I have done. And that has not been countered.
Yeah. Well, the problem is that you're demanding that I show you directly where that happened from millions of years ago. And I'm asking you to live by the own standard that you're setting out, which I think is an unreasonable standard of direct observation. I would like you to show me directly the actual instance of where this was created in the genome, please, because that's the standard you're applying to me.
The result of this act of creation gives us the ability to make testable and falsifiable predictions. That's the gold standard of science. That is what I have done. But you, Mark, have not provided any real testable and falsifiable predictions. You have not presented an answer to really any of my challenges, any of my responses. So I'll ask you this. What testable and falsifiable predictions can you make on endogenous retroviruses?
Yeah, so we've done Phoenix Rising. We've done the paper that Grayson's talking about. There's been other experiments done where they have activated these so-called, you know, inactive viruses that become viruses again. Like, so and it was predicted, the testable prediction was that these would have the structure of a virus if they were indeed viruses that infected. Because don't forget that we didn't, like, it wasn't the creationists that found this. The creationists didn't go, oh, well, I think that God has inserted viruses, so we're going to test the DNA and find them. That's not what they did. It was actually hypothesized by somebody in science, in evolution, to say, hey, we think that viruses have inserted themselves into human DNA, tested the DNA and found them. So it wasn't creationists doing any of this. And do note that creationists haven't discovered any of this stuff. They're relying on the evolutionary scientists and geneticists that came along and found it, and then misrepresent their work to fit with their God paradigm. That's all they're doing.
Both C Science and Dustin would like to ask you and Grayson questions. I'm going to let C Science go first, if that's
well.
Actually,
Just
Stephen, if I could, if I could jump in real quick, because
go ahead, Donny.
It's almost 11:30. I said I had till 11, but this has been so fun. I went an extra 30 minutes.
Yeah,
let me just respond briefly to what he said and then hand it over to C Science and Dustin. If you
go ahead.
So just to leave no stone unturned, can you share my screen real quick? It's just a basic image. You can find it in you can find a diagram like this in many papers. And so yeah, this is the structure of your ERV. Okay, your LTRs on either side with your host DNA, your GAG, your pol and ENV components. Okay, and so with Mark's answer there, I didn't see anything that's future in terms of being testable and falsifiable. You mentioned the Phoenix Rising experiment. Well, this comes down to the question, what is the origin of retroviruses in general? And so this is a much bigger debate. There's a paradox, a chicken and egg problem for the evolutionists, because retroviruses actually require hosts to replicate. So the question to the evolutionists, what came first, the host or the retrovirus? Well, if the retrovirus came first, remember, it requires a host to actually replicate in the first place. Okay, it integrates, it's made of RNA genetic material, and it can perform reverse transcription and reverse, basically. But the creationists would argue that you have host genomes, okay, with these ERVs front loaded into host genomes, and from there you have what's called the escapee hypothesis, which is actually one of the many hypotheses in the conventional literature, where basically retroviruses are harmful retroviruses. That is, they have escaped from host genomes, they have been able to cross species where they can burn hot and fast and lead to disease. And so that makes sense. Okay, so this goes back to the Phoenix Rising experiment, where they resurrected a dead virus. Yeah, we agree. This is agnostic. Okay, they resurrected a dead virus from a host genome. Well, if viruses originated from a host genome, if retroviruses can come from pre-existing endogenous retroviruses that are beneficial, then yeah, of course you would be able to resurrect those from host genomes. And so this doesn't answer the question. This is agnostic. This isn't an actual prediction that is testable, that is falsifiable. I am saying in the future, based on the design hypothesis, we're going to find many more functional roles for these ERVs. That 80% genomic activity, we're going to find within that activity, 80% of the genome, that those ERVs and ERV-like sequences, this is not just noise, this is not just spurious, but this is real activity. This is the trajectory. It favors genome-wide functionality. This is future. This is the gold standard, because we don't know yet what exactly all of these ERVs are doing, but we'll find out in the future when we start performing those experiments. That's something that's testable in the future, but I haven't seen Mark provide that yet, and we'll let him have the last word if he'd like to, and try answering that again. Give me a future prediction that we can test and that is falsifiable on endogenous retroviruses, not something that's agnostic like the Phoenix virus experiment. Go ahead.
Yeah, so all we have to do is just watch the viruses and see how they mutate. That's all we've got to do. We're already tracking their genetic structure. If their genetic structure changes and either a virus becomes malignant again, or if a virus that was malignant to have some sort of benefit for the organism, your thing's sunk. And I think that a virus will mutate, just like we see in the AIDS virus, to provide antiviral load to that organism, because that's how ERVs function. And I think they will show other things as well as time goes on. So get ready for that, Donny, because if that happens, your argument is 100% sunk. And as we understand more and more about retroviruses sequences, as we understand their genetic sequence more and more, and we track them more and more, you're going to have a lot of explaining to do if that happens, because you're basically saying, hey, they were created here, they jumped outside, they were created, even though you can't show in any way that they were created in the cell rather than being in a transmission vector from outside. You can't show that. You're just asserting it, that they were created there. Asked you to show me, you can't do it. If they mutate outside and continue to mutate outside as exogenous retroviruses or as endogenous retroviruses, your entire argument is sunk. So, but I predict that will happen. Now, I'm happy to, if people got questions for me, I'm happy to answer them. It has been 2.5 hours, but
yeah, I appreciate that, Mark. I'll honor that, give you the final word. We'll continue this discussion for sure on ERVs. And okay, yeah, sure. That was a lot of fun. I appreciate Stephen. I appreciate you having me on.
Thanks, Donny.
And the 3 hours has flown by. It's been a lot of fun.